of the greatest scientific detective hunts of all time. Specimens from the top layer of the Earth's surface from every portion of the globe were shipped to the Brooklyn laboratories in the hope of discovering a new antibiotic drug to conquer man's diseases. 100,000 samples of soil to be scientifically searched for a life-saving organism.
I worked on many different organisms and their physiology. We made penicillin, we made streptomycin. We had a glorious number of years, maybe 20 years, with new antibiotics being discovered, but then in about 1960 it slowed down to where it's almost impossible today to see the development of new antibiotics.
Well, this antibiotic field is getting to be really something these days. The antibiotics seem likely to become our most important weapons against many diseases, and actually, as more and more of them are discovered, it seems quite likely that in time we may actually be able to control most of the infectious diseases that plague mankind with them.
Well, perhaps the first thing we should do then is find out just what is an antibiotic.
An antibiotic is an organic compound which has the ability to inhibit certain organisms in their growth or kill them. All sorts of medicine come from these simple compounds that are made by microbes, and they're found in soils all over the world.
Microbes have to have not only food but space in order to live and to grow and to reproduce. To defend their rights, they secrete chemical substances which are capable of making the environment untenable for their
Sort of a chemical warfare among the microbes.
They're fighting all the time in nature. Let us say an organism is faced with an antibiotic and it will die unless it can convert itself to something that will be resistant to the antibiotic.
Organisms have many ways to develop resistance. One is they can block the compound from entering the cell. They can also develop pumps, which, if the compound gets in, pumps the drug out. They can also destroy the antibiotics. They can change the target so the antibiotic can't bind anymore, and therefore you get resistance. But the few that become resistant and are healthy can then take over and resist the antibiotic.
But each time the population of organisms became resistant to the antibiotic, industry had to come up with a new compound, and pretty soon that whole thing ran out. It just was not simple anymore.
The conclusions that were being drawn were we're not finding anything new. And it's kind of, the world is redundant, and we're isolating the same things and same compounds, and therefore let's move on to another approach. Let's stay away from nature a lot.
When people think about pharmaceutical research and development, they think about drug discovery. But really, I think that's a bit of a misnomer, because drugs are not simply found. Drugs are invented.
Nature has been very good to us up to this point, but after years, and I don't blame the companies, after years of pouring millions of dollars into this effort, they weren't getting the return at the end. I think the CDC said in 2013, 23,000 people in the United States died of resistant infections, and more than 2 million had resistant infections. It's a big problem, and if the resistance spreads even further, it will become a bigger problem.
Natural organisms not growing in the laboratory. That's the main problem. Because just by taking a soil and putting a needle in it and plating it out, only 1 in 100 organisms will grow.
We can look at a soil sample or a marine sample or a plant and see that there's much more biological diversity there, but we can't isolate the living organism from that. They may need a lot more interesting signals from the environment they're in that we have no idea about, that are not in that petri dish.
It took 150 years to cultivate 1% of microorganisms by varying composition of the media and cultivation conditions and so forth. What will it take to cultivate another percent? And the answer is not particularly optimistic, because it sounds like another 150 years. A thought occurred that we don't have to manipulate growth media to grow microorganisms, because everyone knows actually where the necessary conditions occur. The next step was to visualize a device. The overall design was clearly simple, or could be simple. I tried piecing together various parts that I was familiar with. Those couple of years of some errors and trials resulted in a different device that we eventually called isolation chip, or iChip. It's a plastic plate with many small through holes. So microorganisms go into those through holes. Membranes are placed on both sides, and then it goes back to the dirt from where the microorganism came from, allowing this dirt to feed the microorganisms, let them grow, form colonies, and after a period of incubation, you disassemble, and you have access to microorganisms you just grew without knowing why. When we got our first data using this device, the increase in microbial recovery was 30,000%. It wasn't 1, it wasn't 10, it wasn't 100%, it was 30,000. So even if that was some sort of a fluke, and the next trial wouldn't bring you 30,000%, it was clear that the increase was significant.
What we have here are microorganisms that have been isolated from the iChip, from soil. And you see a diversity of microorganisms here. You can tell that based off of color.
It's true that the number of colonies here exceeded the number of colonies in Petri dish by a very significant factor. But we didn't know if those many new colonies were actually representing new species.
I know Teixobactin hit the news recently, and I knew of it, and it's a very exciting compound. It's a long way from the market, but it's an exciting compound.
There are several very cool aspects of this particular molecule. Typically, microbes in the lab will develop resistance to a trial antibiotic pretty quickly, in an overnight culture. That didn't happen with Teixobactin. Resistance did not arise in a space of several weeks of incubation in the presence of Teixobactin, which tells you that the frequency of resistance is very low.
The real story behind that antibiotic, although the antibiotic itself is really interesting, as a microbiologist, what I find most fascinating is the organism that it comes from, which is this novel beta-proteome microorganisms that no one has ever cultured before. And it came out of a device just like this, out of an iChip.
I do not think that iChip specifically selects for microorganisms that specifically makes wonder drugs. That's not the case. What this technology does allow is to sample microorganisms from the pool that previously has not been sampled.
It's kind of like the Wild West of microbiology, that you're wrangling these microbes that have never been cultivated or domesticated in the lab. And some percentage of those, we're betting, are going to make novel antibiotics.
This antimicrobial action is a complex one, which even today is not fully understood.
We don't know why the majority of microorganisms do not respond to the cultivation conditions we can provide. There may be a multitude of reasons, but we don't know what they are. We're simply beating the odds of discovery, and we're beating these odds by orders of magnitude.
If you want to be right, just say it won't work in our business, because that's the nature of the business. We fail more than 99% of the time to bring something from discovery to the end. But we succeed sometimes. It has a lot of value. So why we do it.
I think this iChip business is going to make companies think twice about being out of the antibiotic field. If they can isolate these organisms from nature that weren't isolatable by the means that they were doing, they might come back and take a look at it. I don't know. I would hope so.
We cannot learn about microbiology, about microbes, how they live, how they interact, how they influence the biosphere, what properties are useful for us. We just can't unless we have access to the remaining 99%.
What new discoveries still lie locked in the soil? What possible cures for diseases as yet untouched? Pressing steadily forward, scientists and physicians of vision and experience may soon make the next great contribution to the age of antibiotics.